Hello, and welcome to the Metabolic Classroom. I'm Dr. Ben Bikman, a biomedical scientist and professor of cell biology. Today, we're exploring the intricate metabolic regulation of autophagy. We'll delve into what autophagy is, its significance in health, why you should care at all about it, how hormones influence it, as well as how various interventions can modulate autophagy, and in some instances, how this might be leveraged to extend lifespan, or why even explore autophagy in the context of longevity. That is, after all, what's really made longevity kind of famous. We'll also examine the crucial role of hormones, in particular the petite peptide insulin, in regulating autophagy and discuss the impact of diets that adjust insulin, like ketogenic and low carbohydrate diets, and even the direct effect of ketones on this cellular process. By the end of the lecture, my hope is that you'll have a pretty comprehensive, a good understanding of how autophagy works, what it is, and how you might be able to take advantage of it for some healthy longevity, with a healthy dose of appreciation that this is a very young, new, unclear field. Now, before I go any further, I want to remind you about the launch of my new site, benbikman.com. At that site, you can subscribe to become an insider at two levels, a basic insider or a pro insider. Each has some benefits that I know you're going to enjoy if you are interested in staying up to date on the latest science in the realm of metabolic health. To learn more, of course, please just go to benbikman.com. Now, as we get started, what is autophagy? Autophagy is derived from Greek for self-eating. This is a fundamental cellular process where cells will degrade, break down their own components, and use those building blocks to build up something else. This process is essential for a cell maintaining its normal homeostasis, so maintaining normal function. The cell uses autophagy to replace damaged organelles. That's its most famous role. Now you're thinking, well, what are organelles? That's a little bit of cell biology here. Organelles are basically all of the components of the cell. This includes things like the nucleus, the mitochondria, the peroxisomes, the ribosomes, all of these different characters within the cell. Over time, they get damaged or old, and autophagy allows them to be replaced. And there's one other component to this that I'll mention in a moment, but also such things as faulty proteins or misfolded proteins that need to get cleared out lest they start to cause damage, and a handful of other cellular debris and stuff that needs to be removed. Now, the essential organelle that I just alluded to a moment ago that's involved in this process is called the lysosome. The lysosome is often referred to as the cell's recycling center. It contains very specific and specialized enzymes that are capable of breaking down a wide variety of these molecules, including lipids, proteins, and various carbohydrates. And that's, of course, the stuff that these organelles are made of as well. Now, by degrading the damaged or unnecessary components within the cell, the lysosomes play a very important role in maintaining the health of the cell. And then, as I noted earlier, once the lysosome has engulfed this organelle and kind of targeted it for destruction or breakdown, it will then give back those building blocks to the rest of the cell. That's why I use the word recycled. So it gives those building blocks back to the cell to be used for the production of new molecules, or in some instances for creating energy, creating ATP from those molecules. Like, for example, the lysosome can pull in a cholesterol ester and break down the cholesterol into its sort of component parts, as well as releasing the fatty acid from it. And that fatty acid can go and be used for energy. Now, suffice it to say, all of this is simply to help the cell survive and work well. Now, so what? So autophagy is a fundamental principle in cell biology, but what about the broader context of physiology? How do the ideas of cell biology with autophagy fit within the context of the human body? In other words, why should you care at all about this? Well, some of it's becoming already clear. There are a handful of functions where autophagy plays an important part, like, for example, cellular quality control. By degrading and recycling the damaged parts of the cell, the organelles and proteins, autophagy is able to prevent the accumulation of these waste products that are taking up space that over time can impair the function of the cell and potentially lead to some diseases. And I'll get to some disease examples in just a moment. I also just a moment mentioned energy levels. And of course, during periods of any kind of scarcity, including fasting, of course, and then starvation in extreme instances, autophagy can provide these building blocks and the energy for the cell to continue to survive despite the relative deprivation. Now, importantly, I mentioned that you can break down molecules to not only get bigger molecules, to not only get amino acids that can be recycled to synthesize new things or be used for energy. Some amino acids are energetic, if you will, or can feed into the citrate cycle and be used to generate ATP. But you also can get a lot of fatty acids as well. And of course, those fatty acids, as you're breaking down any big lipids, and that includes glycerolipids like triglycerides, diacylglycerols. It includes sphingolipids like ceramides or sphingomyelin. As all of those are broken down, you get that free fatty acid, which can then, of course, just go to the mitochondria and be used through with beta oxidation in the citrate cycle or the Krebs cycle to create an abundance of ATP. All of this simply means that the recycling mechanism of autophagy is very crucial during any time of nutrient deprivation. So if calories have generally stopped coming in or insufficient to support the body, autophagy is one of the ways in which any given cell is going to ensure that it's getting enough energy from itself. Now, a moment ago I mentioned disease. Proper autophagic activity, that's the weird adjective form of autophagy, autophagic. Proper autophagic activity is important for preventing various diseases. In particular, the most relevant is probably the neurodegenerative disorders like Parkinson's or Alzheimer's disease. There is substantial strong convincing evidence that autophagy is beneficial in those processes. Also with atherosclerosis, autophagy appears to be beneficial, and even, of course, cancer. If you can tell a cell to get rid of stuff, it can help control cancer. Additionally, and very of great interest to a scientist like me, autophagy or the lack of it can be a relevant factor in obesity and its associated problems. Now, as your temporary professor, let me just go on that topic for just a few minutes, because there's a pretty interesting body of literature that has explored the connection between autophagy and not only just fat gain, but also, as I mentioned, the consequences of that fat. So there are studies, and I include several in the show notes, that suggest that excessive fat accumulation impairs the autophagic activity in adipose tissue. So for example, in obese mice, if autophagy is inhibited in the adipose tissue, it increases the inflammation in that fat tissue, which of course is a hallmark of metabolic dysfunction, something that I've discussed abundantly in the past. One of the problems with fat cells that get too big, or the hypertrophic fat cells, is that they become very pro-inflammatory, releasing a host of pro-inflammatory cytokines into the body. And as systemic inflammation goes up, insulin resistance follows. And of course, inflammation itself will have a host of problems contributing to things like, say, cardiovascular disease. But in those same animal models, if you can restore autophagy through genetic or pharmacological interventions, you're able to reduce the inflammation, you improve the blood flow and function of the fat tissue, and no surprise, you enhance insulin sensitivity. So collectively, these findings highlight autophagy as a potential mechanism linking obesity to some of the metabolic complications associated with it. Now, in humans, you can't get quite as mechanistic in humans as you can in rodents. That's generally always the case. But the evidence is emerging, but it is nevertheless promising. As much as there is a bit of a gap in the human evidence, it still is interesting. There are some observational studies that indicate that autophagic markers in adipose tissue are altered in people with obesity compared to those that are lean. Now, just by way of clarification and a teaching principle, when I note that a study or the evidence is observational, this means that you, in this case, you had obese or lean individuals and you simply conduct a fat biopsy and then look at the differences in the adipose tissue. So there's no intervention. You're just observing a phenomenon, in this case, between two populations of humans. Now, in this particular study, looking at the adipose tissue from obese and lean individuals, they found that markers of autophagy, and I'll mention a couple now, like LC3 and ATG5, you can look both of these up. These are both protein components in the autophagy pathway. So the overall process of activating or turning on autophagy to activate the lysosome to start identifying and breaking down the necessary parts of the cell. So again, these markers of autophagy like LC3 and ATG5 were elevated in the omental or the visceral fat compared to subcutaneous fat. And this upregulation was particularly pronounced in the obese participants and was very strongly linked to the degree of visceral fat accumulation and adipocyte hypertrophy. So the findings suggest that autophagy is active in visceral fat during obesity. And it could be as a compensatory mechanism to try to manage the stress caused by all of the excess fat, particularly the hypertrophic fat. But it doesn't necessarily, it could also, the authors speculate, be evidence of a dysregulated autophagy process, which might exacerbate the overall issues of inflammation that comes from the hypertrophic fat cells. So suffice it to say, as unclear as the evidence is in humans, there are differences as the fat tissue starts to expand, but the specific degree to which the autophagy is mediating or complicating processes is still a little unclear, and in that case, we have to rely more on the animal evidence. Now, let's transition, while still including some metabolic focus here. I would be remiss as your temporary professor if I didn't include some discussion on longevity. That is, after all, how most people have even become familiar with the word autophagy. It is really a function of the rise or the explosion in longevity as both an area of research and an area of kind of hype social media influencers. Now, nevertheless, most people are interested in longevity. I certainly want to live a long, healthy life. And so let's take just a few minutes to discuss the connection between autophagy and longevity. And based on everything I've said, it's easy to see how autophagy is relevant in the realm of aging. After all, if autophagy is relevant to complications of obesity or neurodegenerative diseases or cardiovascular health, clearly those are diseases associated with aging. And so the more we can control them, the more we are aging well. So evidence does suggest that autophagic activity declines with age, leading to the accumulation of damaged cellular components, which we could say is a level of aging at the cell itself. If you can't get rid of damaged components of the cell is, we could say, getting old. So enhancing autophagy, no surprise, has been shown to increase lifespan and improve health span in a handful of organisms. Now, I'm using that word very deliberately. So like, for example, animal studies. I'd earlier mentioned an autophagy protein called ATG5. In mice, the genetic overexpression of the autophagy-related gene ATG5 has been shown to enhance autophagy, no surprise, which in this case, and this is, of course, a global issue in this case, or a global phenomenon where everywhere the ATG5 is going to be acting a little more rapidly than normal or more active. But this, in these animals, led to an extended lifespan and generally improved health markers, including improved insulin sensitivity and overall motor function. So they moved better as well, which suggests a benefit to both muscle and neural regulation, because movement is, of course, a combination of those two things. Now, of course, there is some pharmacological interventions that have been studied, and I'll only mention one, a famous one, or infamous if I had my way, rapamycin. Rapamycin, as you know, and I have had an entire metabolic classroom on this topic, is an mTOR inhibitor. That's what's made it famous. That's what's made it famous. So mTOR is an important cellular protein complex that only wants to build. And I'm going to come back to this idea in just a minute. But mTOR wants to build up molecules, whereas autophagy wants to break things down. So no surprise, if mTOR is turned on, autophagy is turned off. In contrast, if mTOR is turned off or inhibited with rapamycin, autophagy would be turned on. So by inhibiting mTOR, rapamycin would, of course, promote autophagy, which can, no surprise, contribute to an improved health span. Now, I strongly encourage you to watch my previous metabolic classroom on this topic. I spoke about it abundantly, attempting to highlight some of the positives, which are all most people ever hear, but of course, very deliberately and I hope convincingly go over the negatives, because there are substantial considerations with the use of this drug by biohackers and others who believe that they're going to live longer. Importantly, all of the rapamycin evidence that suggests it promotes longevity, all of it is based on animal or insect studies. And in this case, these are organisms who have the benefit of living in utterly germ-free, sterile facilities, so they don't need to worry about any sort of immune compromised effects, which rapamycin does, compromise the immune system. And they're all genetically identical. So these are people, these are not people, organisms, the animals or insects, that are all exactly the same. Well, suffice it to say, humans are not. So it's no surprise that someone may report anecdotally feeling great on rapamycin while others are experiencing all of the complications that come with it, including compromised immune system, including compromised muscle recovery and athletic performance, etc. So please see that earlier metabolic classroom to understand some of the considerations with rapamycin, that it's not all rosy, that as much as autophagy is a very beneficial process, in this case we could say it's a little too much of a good thing, which is so often the case when it comes to drugs. All right, now, me discussing mTOR and mentioning diet, at least in the introduction, it allows me to introduce a character into the story that you've heard me discuss before, and indeed it has been difficult for me not to mention it yet. After all, few things influence mTOR more than the humble hormone, or what I said earlier, the petite peptide, insulin. Insulin plays a central role in regulating autophagy because it plays a central role in signaling a nutrient level. And in particular, when insulin goes up, the body transitions into what I like to refer to, by quoting other famous physiologists, the fed state. And the fed state is a state of storage and growth. That is anabolic. So insulin is one of the most powerful activators of anabolic or building processes. When insulin levels rise, such as after a carbohydrate-rich meal, so eating refined starches and sugars, that is going to spike insulin considerably. And that alone is not a bad thing, but as it happens too much, it becomes problematic. But suffice it to say, this signals to the body, to the cells, that nutrients are plentiful and that it's time to grow. It's time to build molecules or become anabolic. This, of course, relies on protein mediators like mTOR. But mTOR is anathema to autophagy because autophagy, insulin and mTOR wants anabolism to predominate, while autophagy is the ultimate catabolic process. So in this context, insulin encourages the cell to focus on anabolic processes like protein synthesis or glycogen storage or lipid synthesis, but namely or collectively prioritizing growth and repair over the catabolic breakdown of these molecules and recycling of organelles. Now, these opposing forces, anabolism promoted by insulin and catabolism mediated through autophagy, highlight the balance that the cell has to maintain to function optimally. When insulin levels are persistently elevated, like you have as both cause and consequence of insulin resistance in type 2 diabetes, autophagy can remain chronically suppressed. You don't want that off all the time. This can lead to the accumulation of the damaged organelles and proteins and ultimately contribute to cellular dysfunction. We want to balance insulin levels. So if insulin gets spiked, it's up and then it's down. We need to have this balance of periodic activation of autophagy or catabolic processes, allowing the cell to clean itself, but we balance that with necessary anabolic activities. Okay, now talking about insulin prompts me to transition into low carbohydrate diets or ketogenic diets, and then how they influence autophagy, because there is growing evidence on this that's pretty compelling that I think you'll find interesting. Of course, if a diet is ketogenic, that means by necessity it is a diet that has lowered insulin. Insulin is a powerful inhibitor of ketogenesis. So if a body is ketogenic, making its own ketones, then insulin must be low. And for insulin to be low, you're either fasting a lot or you are restricting carbohydrates. So a low carbohydrate diet is generally, if carbs go low enough, going to be the ketogenic diet. Now, studies do indicate that ketogenic diets can enhance autophagic activity in certain tissues, including the brain, which can explain some of the benefit that people see with neurological disorders while adhering to a ketogenic diet. In fact, for instance, research has demonstrated that the treatment with a ketogenic diet does, in fact, increase the number of autophagic structures. So this evidence, the cellular evidence of autophagy being turned on in the hippocampus and the cerebral cortex in rodent models. So that very, very strong, very convincing evidence that a ketogenic diet is, in fact, promoting autophagy in the central nervous system, which, of course, is going to be very helpful with a handful of neurological disorders. Even further, I earlier mentioned kind of bad proteins or misfolded proteins. Misfolded proteins are a feature of cells, just normal cell biology, but as they accumulate, they can activate processes like inflammation through another process called ER, or endoplasmic reticulum stress. You can look that up to learn more, ER stress and inflammation. But also certain neurodegenerative disorders, like even Parkinson's, have been are believed to be influenced by misfolded proteins. Interestingly, beta-hydroxybutyrate, which is the primary ketone in the body, can support health by addressing this. So firstly, BHB directly enhances autophagy, a process, of course, this overall process of breaking down things, including misfolded proteins. So BHB is a direct activator of this autophagy pathway. As a result, no surprise that as BHB goes up, misfolded protein content within the cell goes down. One of the ways also this happens, separately from just activating the autophagy pathways, is that BHB can change the solubility of the misfolded proteins, which helps them actually get into the lysosome in the first place to undergo autophagy. So this is a sort of dual benefit here. Activating the autophagy pathways and then directly enhancing the degree to which misfolded proteins get engulfed by the lysosome to undergo that autophagy. So ultimately, with regards to the ketogenic diet, and to wrap up that particular bit or that particular principle, there are two factors to be aware of when it comes to the ketogenic diet and autophagy. The first, as I mentioned when starting this little bit here, is that if a diet is ketogenic, it is necessarily one that keeps insulin low. When insulin is low, mTOR activation is low, and thus autophagy activity is high. That's a good thing when it comes to just generally wanting autophagy. But also, as mentioned, ketones have a direct effect as well, activating autophagy, and in the case of misfolded proteins, enhancing their kind of targeting by the lysosome. Now, one final note on all of this before I wrap up. You don't want autophagy all the time. Healthy cell biology and, by extension, physiology requires periods of both building up and breaking down. You have to have a balance of the anabolic and the catabolic. Too much catabolic, the body just starts to waste away. All tissue starts to decline. This is one reason why I'm so opposed to the drug activators of autophagy like rapamycin. In that earlier metabolic classroom I did, one of the studies I shared with you by Drummond et al. was a human study that found that those humans on rapamycin had reduced muscle protein synthesis following workouts. It's directly challenging the muscle's ability to recover and build to be bigger and stronger following a workout. That is not a good trade-off. Now, what about a ketogenic diet? Some may think. Because that is a state of low insulin, is it too catabolic? Well, thankfully, the evidence suggests no. That one important thing is that as much as a ketogenic diet is going to be a diet that is generally keeping insulin low, you may think, well, then I'm not going to have any anabolic activation. When I want my muscle to grow and I want mTOR turned on and autophagy turned off, how can I do that in the absence of too little insulin? First of all, there are other hormones that activate that, like IGF-1. That's the insulin-like growth factor, also known as the somatomedin or somatomedins. There are several. And those are produced when growth hormone goes up. Well, guess what happens on a low-carb or ketogenic diet? Growth hormone and IGF-1 can go up. But at the same time, generally when someone's adhering to a low-carb diet, they're eating an abundance of protein. And when you eat protein, that converts into amino acids. And guess what does similar effects to insulin in this case, namely activating mTOR and inhibiting autophagy, allowing for tissue growth? Amino acids. So amino acids become a bit of a redundant mechanism here that allows a person on a low-carb diet who may be worried about their low insulin when it comes to chronic activation of autophagy, but they need not. Amino acids play that role. They play the part that insulin might have in this particular context. All right. In today's lecture, we explored the fascinating process of autophagy, the cell's natural recycling system, and hopefully you've learned a little more about why it's important as a process in maintaining the health of cells and even how it can help promote to longevity. Through the brief lecture, we covered how autophagy can decline with age, how insulin regulates this, how nutrients regulate it, including things like fasting and low-carb diets. Even, of course, I gave a shout out to some of the popular pharmacological regulators, which, as you know, I'm unabashedly opposed to. Autophagy is a healthy process. We want to take advantage of it, but the best way to do so is not through some drug with substantial side effects, but rather by manipulating, I believe, the nutrient signals. Those nutrients that we eat, they are our way of turning autophagy on and off when we want them on and off, because that is what you need. Healthy physiology is a balance of building up and breaking down. All right. Thank you for joining me today on the Metabolic Classroom. I hope the lecture provided you with some actionable insights and at least a deeper appreciation for the metabolic processes that sustain our health. Until next time, more knowledge, better health.